speaker.
Hello, thank you for the opportunity to present today. I'm going to tell you about our work in interpreting cancer genomes. Now, cancer has become really the public health challenge of the 21st century. As we live longer lives, more of our lives are affected by cancer, and it's projected that the cases of cancer will increase by 50% in the next 20 or so years. And I'm sure many of you or your family members or your friends have their lives affected by cancer. And projecting in terms of deaths, it's projected that deaths due to cancer will increase by roughly 60%. Now that's the bad news. The good news is that over the past 30 years, we've learned a lot about what molecular mechanisms lead to cancer. And what we now know is that cancer is caused largely by somatic mutations. These are mutations that develop in our cells after we're born. These mutations arise naturally, just as cells divide and copy their DNA, and they also arise as a result of exposure to various environmental effects like cigarette smoking, UV light, etc. And so over the course of multiple generations, we get many of these mutations. And the other good news is that we've now been able to measure these mutations really in much more efficient ways. And this has been driven by new technologies that allow us to sequence genomes and measure the billions of letters that make up our genomes very, very efficiently. And the cost to sequence a genome, a human or cancer genome, was hundreds of millions of dollars a decade ago, and now is approximately $1,000. So we can use these great technologies to measure these mutations. And the other good news is that we now have some drugs that allow us to treat patients with very particular mutations. So this has led to the suggestion that the new era of cancer therapies will be personalized, whereby we measure the genomes and other molecular measurements of an individual patient's tumor, and we're then able to target the treatment towards those particular molecular profiles. And this is, of course, good and hopeful, and there have been many successes in doing this. However, there are actually many, many challenges, because when we look at an individual patient, we need to take their genome for this one patient, identify the mutations using the technologies that we now have at our disposal, but then we're left with this question of how do we go from the tumor genome of this individual to a particular treatment. And without other knowledge, this becomes a very, very challenging problem. So what we need to do is we need to learn more about the mutations that lead to cancer, and which of the many mutations that accumulate in our genomes over our lifetimes are the ones that might cause cancer, and which are merely just random so-called passenger mutations. So in order to develop such a catalog, several large-scale projects were launched about a decade ago. In the US, there was a project called the Cancer Genome Atlas. There were other such projects in many countries, including China, under the umbrella of the International Cancer Genome Consortium. And what these projects did was use this DNA sequencing technology and other related technologies in order to profile the tumors of thousands of patients. So the US TCGA project measured the genomes of roughly 11,000 patients with 33 different types of cancers using 7 different technologies. And so now with this data, we can then start to identify which of these mutations might be the ones that lead to cancer. And we can do this in a way that allows us to make sense of this large data set. So by taking the genomes from these many thousands of individuals, we can then start to compare them. We can first identify mutations that are present in the tumors, but not present in our normal cells. And then we can start to look across the individual patients and identify the mutations that occur more often than expected by chance. Or perhaps we look in a region of a gene and identify genes that are mutated more often than expected by chance. And these recurrent mutations suggest that they might be cancer-causing. And so this is a paradigm that's been used, and in the past few months, this project, the TCGA, has just concluded, and one of the papers that came out of the analysis of all the data collected to date was the identification of 299 driver genes, genes that were mutated more often than one would predict, and therefore might be ones that are causing cancer. And so this was a great success, but also if we look deeper at the data, it leads us to see that, in fact, these are likely not all of the genes that cause cancer. And in fact, what we see as we look across these genes and we look at how often they're mutated is while there are a handful of genes that are mutated very frequently in cancer, there's a so-called long tail of genes that are rarely mutated. And so by comparing just these even 11,000 genomes, it's very hard for us to go further down into these rare mutations and pick out the ones here in dark green that were known to be cancer-causing genes. So this long tail really challenges a lot of these analyses. And it really points out one of the really key challenges in looking at cancer, and that's that cancer is highly heterogeneous. There's heterogeneity across tumors. When we look across different individuals, we see a large diversity of mutations across tumors. Even within one tumor from one individual, there's great diversity of the cells within that tumor, where different cells have different mutations. So we need to be able to make sense of this great diversity. So when we look across patients, this large diversity of mutations is apparent in this long tail phenomenon, and this is why we have a hard time digging into this long tail. When we look at one tumor and we sequence this tumor with these great technologies we have, we're measuring the tumor in bulk, and we're actually measuring a mixture. So what's happened now is the challenge has shifted not from measuring mutations, measuring tumors, but to interpreting the resulting data. And cancer, in many ways, cancer genomics has really become a problem of computer science, a problem of data analysis, a problem of machine learning and algorithms. And so I wanted to tell you today about two of the projects we've been doing in my group, one to identify recurrent combinations of mutations, and one to look at the evolution of a tumor within one individual. So starting with this inter-tumor heterogeneity, we again are faced with this phenomenon of this long tail of rare mutations. And if we think more deeply about this phenomenon, we think about how biology works and how a cell works, we realize that genes don't work alone, they work together in pathways and networks, as you saw in the previous talk. So rather than interpret this picture one gene at a time and try to identify one gene at a time which ones are the cancer-causing genes, we should instead be looking at aggregate, and we should be looking at the genes and how they interact with each other and try to find these recurrent networks that are mutated more often than expected. And so this is what we can attempt to do, and we can try to take these large data sets and try to find from these large data sets which subnetworks are mutated more than expected by chance. So if we take the network of all gene interactions in the cell and we take mutation data from thousands of patients, if it's true that these cancer cells have mutated particular networks, then we should be able to find pieces of this network that are mutated more often than expected by chance. So we need to find these groups of genes that are working together to drive cancer development. This turns out to be actually quite a challenging problem because these networks are highly interconnected and they're highly variable. Some genes interact with many other genes, some interact with few genes, and they tend to have this phenomenon of what's called being a small world network. So it doesn't take very long to go from one gene to another in this network. So we need very robust and careful algorithms in order to do this reliably. And within my group, several years ago, we developed an algorithm that attempts to take this gene network information, this mutation data, and identify which parts of the network were mutated more often than expected by chance. And the way we can think about this problem is that we can think of the initial data we have on how often a gene is mutated as a source of heat on that node. We assign a heat value to that gene according to how often it's mutated. And then we let that heat diffuse over the network for a certain length of time. And when we've let that heat diffuse, what we'll sort of see is that some parts of the network are hotter than other parts. And so then we can identify those hot spots on the network. And those hot spots on the network are interconnected genes that together are mutated more often than expected by chance. So we developed this algorithm. It was called HotNet, and it was used in about a third of the cancer types that were analyzed by this TCGA project. And just to show you one such analysis, here an analysis of 12 different cancer types, 3,000 samples, and we identified 16 different subnetworks, here shown arranged around the circle according to the cancer types in which they're mutated, that really rediscovered a lot of the networks that were known, but also suggested some new networks and complexes of proteins that were working together to cause cancer. So if we just zoom in on one of these networks, we see here just a piece of a larger network with 30 other genes. And right here, this gene in red, this very hot gene, is the p53 gene, one of the most famous cancer genes there is. So it's not surprising we find that, but we find actually that many genes interacting with p53 are also, although on their own, not mutated very frequently, are very cold here in blue, but they're sort of picked out by the algorithm according to their interactions with p53. And in fact, just to show an example of one of these, CHD8, this is a gene that was mutated in only 54 of the 3,000 samples. And what we sort of see is this pattern of rare mutations across these 34 samples from different cancer types shown in different colors. These little bars here indicate that these mutations damage the gene, prevent it from doing its function. And right here in green, we see this pattern of damaging mutations in glioblastoma, a brain tumor. And a year or so after we published this analysis, it was shown that the CHD8 gene is actually involved in another type of brain cancer, low-grade glioma. So this is an analysis that really, though, only looks at the protein coding genes. And the protein coding genes are only 2% of our genome. So what about the other 98% of our genome? And how can we analyze these non-coding regions? So we have recently worked with another project, again, part of this ICGC, which sequenced these whole genomes, and we tried to find whether or not we could find these non-coding mutations. And very quickly, when we look one gene at a time, we see that this long tail phenomenon in the protein coding genes is apparent, although in dark green, we see the known genes. But in the non-coding region, the long tail is even longer. And there's very few strong signals in the data, and we really need to dig deeper into this long tail of rare mutations. And this is where this network analysis will help. And so I co-led the network analysis for this PCOG project, as it was called, where we put together the coding mutations that were found, the non-coding mutations. We analyzed the data separately together. We analyzed it with our HotNet algorithm, as well as 6 other algorithms that look at the data in aggregate, and we predicted 87 genes that were sort of significantly mutated in their protein coding regions, but another 93 genes that were significantly mutated in their non-coding regions. And so what this allows us to do is start digging deeper into this long tail. So in dark green, you see the genes that were predicted by our method. So all the dark green genes here in the, that are currently mutated are predicted. In blue are genes that were previously known to be implicated in cancer. So a lot of these rarely mutated genes that we find in coding regions were already known to be implicated in cancer. But in the non-coding regions where there really isn't much signal up here, we can really go into this long tail and find many new genes that are mutated in their non-coding regions and likely to drive cancer. We can also look at the network level. Is it mutations in protein coding genes or mutations in non-coding regions of the genome that seem to be driving cancer? And we, of course, find that it's both, but seemingly with different contributions of coding and non-coding mutations, depending on the biological process. So we find that there are lots of non-coding mutations in genes that are associated with developmental processes, with things like RNA splicing, whereas genes that are involved in sort of core proliferation pathways seem to mostly be driven by coding mutations. These are the ones that have been largely known from previous studies. So next, we want to now look deeper at the tumor of a single individual and look at this heterogeneity within a single tumor. So when we sequence a tumor, this tumor is a collection of cells that were the result of this process of mutations accumulating over time. And this is a mixture of cells, some normal cells that don't really have somatic mutations of interest, and all the somatic mutations that have developed in this tumor. And when we sequence the tumor in bulk, what we obtain are measurements of these mutations and their estimated frequency. But what we'd really like to know are what exactly were the tumor cells and the mutations that they contained, and what proportions were each of these cells in the mixture. So what we need to do is, from this data that is really a composition of a mixture of everything, separate out the components of this mixture. So can we pull apart the components from the mixed measurement? Now this is sort of idea of separating out the components from a mixture is actually a sort of well-known problem with lots of variations in different fields. In fact, maybe the most famous and perhaps the most interesting is the so-called cocktail party problem. Maybe at future forum you'll get to to join a cocktail party, and when you're at a cocktail party, there's many conversations going on, but somehow our brains are able to focus in on the conversation of interest. Or sometimes we might get distracted by the person in front of us, and we might eavesdrop on another conversation. And our brains can modulate which conversation we'd like to hear. So we have a very good way of pulling apart this mixed signal that's presented to us into these component conversations. And what allows our brain to do this, so it's very complicated, but one of the things is that there's structure to the data. Conversation is spoken in a language, and we can use this sort of structure to the data to try to pull it apart into components. And our cancer data is also structured because these mutations that we measure are not random, and these cells that we measure are not random. They're a result of this evolutionary process that led to the tumor. As these mutations accumulated over time, later mutations were survived and had some advantage in terms of growth over earlier mutations. And so we have this structure, this evolutionary structure of the data, and we can use this structure to try to help us solve this problem. Moreover, finding this evolutionary structure is also very useful for learning more about the evolution of tumors, because it's really the evolution of tumors that really drives some of the things like resistance to treatment, because the tumor can continue to evolve and develop new mutations that confer resistance. It's related, this evolutionary process, with the aggressiveness of the tumors. They're related to a tumor relapsing or metastasizing and colonizing different sites in the body. So understanding this evolutionary process is very, very important. And like any evolutionary process, the evolutionary process of a tumor can be described as a tree, a phylogenetic tree, a family tree. And in evolutionary biology, there are many algorithms that have been developed to take data from different individual genomes, here different species, and we measure whether or not these species have different mutations in their genomes. And then we can take this data at the present time, and we can recover the past. We can infer who is related to who and who are the closest relatives by looking at the similarities in their genomes and building this family tree back in time. And this is what we like to do in cancer. Unfortunately, our data is not just measurements at the present time individually. It's all mixed together. So what we have are not the DNA sequences of individual species. Instead, we have sort of mixtures of species, and we have our measurements are the DNA of different continents. So could we, from these mixtures, of DNA of, say, South America and Australia and North America, recover, again, this relationship between the genes. So this is our separation into components problem. And we can do this in various ways using different algorithms, and I'm going to show you sort of one result of one of our algorithms and its application to a kidney cancer. So here is this kidney cancer that was chopped up into individual regions. 9 different regions from this kidney cancer were sequenced. And in the original publication, a tree was published that shows here's the original germline, the normal genome, many mutations accumulating over this branch, and then mutations that distinguish each of these different regions of this tumor. This is a so-called phylogenetic tree. What we uncover is that each of these regions is not a single cell. It itself is a collection of cells, and each of those is a mixture. So in fact, what we can see is that the tree is not a tree on regions, but it's a tree on the mutations that are present, and each region is a mixture of the different cells with different mutations at those times. So we see each of these different regions being a mixture of these different leaves of the tree. So this looks, very complicated in this sort of picture, but using these algorithmic analyses of cancer, we don't often just get more complexity. Sometimes we actually simplify the picture considerably. And so for an example of that, another issue that often arises in looking at cancer and its evolution is in understanding how cancer metastasizes and colonizes different organs. And now with the ability to sequence more reliably, we can start to sequence the primary tumor and metastases from the same patient. Here's one such example of a patient with a metastatic breast cancer whose tumor had unfortunately metastasized to multiple different organs. And in the original analysis, what they tried to do was recover the history of how these migrations had occurred. Where had the cells from the primary breast tumor gone and colonized, and in what order, and did these colonizations happen one time or many times? And what they uncovered was a very complicated picture where there were 14 different migration events from cells, tumor cells, leaving the breast, going to the lung, going to the rib, from the rib to the lung again, multiple times to the brain, multiple times to the kidney. And this so-called idea that a metastatic site was seeded by multiple migrations from other different tumor sites. If you look more closely at the data, and if you analyze it, being well aware of this fact that each of these samples is a mixture, and if you solve this cocktail party type problem that I described, what you can actually uncover is that the data is equally consistent with a much simpler story where the primary breast tumor had cells that first migrated to the lung, and then in single events seeded the metastatic tumors at each of the individual sites. And we were able to reconstruct this picture using an algorithm that was published just earlier this year called Machina, work of my postdoc and graduate student listed there. So what I hope I've been able to show you is that really with these challenging data sets that we get from large-scale sequencing of tumors, the challenges have shifted not from generation of the data, but to analysis of the data. And there are many subtleties in the data because of the complexity of the heterogeneity of tumors that need to be addressed. And as we now think about treating tumors, we have to think not of treating a tumor as a single mass, but we have to think of treating a tumor as an evolving ecosystem. And so, it's not just one targeted treatment or one type of immunotherapy that might kill all of the cells in the tumor. It might be necessary, and in fact, it will be required to use some combination of drugs that together sort of take care of the multiple cell types within a tumor and prevent the tumor from evolving resistance to the single drug treatment that we might give it. Now, this seems very, very challenging, and indeed it will be challenging, but we've actually, in medicine, has done this before. And here's another disease that you'll see increased dramatically in incidence and deaths, and then suddenly dropped off. I don't know if anyone knows what disease this is from looking at the dates here. This is the story of HIV. And in HIV, it was really and continues to be combination therapies that are used, multiple drugs that really target this highly mutating, highly evolving virus and keep it in check. And so what we will need to do in cancer is develop these combination therapies, but unfortunately there's not one single virus because cancers arise independently in each individual, so we're going to have to develop these personalized drug combinations. We're going to have to solve the combination therapy problem separately for each individual, which sounds hard, but fortunately for us, there's a lot of data that's now become available. There's the ability to measure an individual's tumor with really high accuracy, much more cheaply than we could do even just a few years ago. And there are dramatic advances in machine learning and algorithms that will allow us, in some way, to develop these biologically informed intelligent systems that allow us to, for an individual, identify not only the mutations that might be driving their cancer, but the combination of mutations that will help keep that tumor in check and help that person live a long and productive life. So with that, I'll conclude and just acknowledge the individuals that contribute to this work, members of my group on the algorithmic